PCN was detected in 2006 in a tear dirt sample from a grading facility in Blackfoot. And later that same year, it was traced back to 7 infested fields in the Shelley, Idaho area. Since then, 11 years later, through efforts of the Pale Cyst Nematode regulatory program, the infestation has been contained to a small area with an 8.5-mile radius that spans portions of northern Bingham and southern Bonneville counties. So I'll just tell you a little bit about the pest if you're not familiar with it. It's a plant parasite that infects the roots of certain plants that can host the pest. PCN has a very narrow host range. It can only feed on plants, solanaceous plants such as potato, tomato, eggplant, some peppers, and some weeds. Potato, though, appears to be PCN's favorite food because it can reproduce more effectively on potato plants than any other host. Most of the PCN life cycle is spent in dormancy. The nematode eggs are inside of a cyst. The cyst is actually the dead body of the female worm after reproduction. And the cysts are extremely small, just barely visible to the naked eye. If you were to hold one, you'd feel it in between your fingers, like just barely like a grain of salt. They have a lot of potential packed in a little space, though. They can contain up to 600 eggs, so there's a lot of reproductory potential there. What makes them very difficult to detect and kill is that they can remain dormant in soil for 30, or some reports even suggest 40 years in soil without the presence of a host crop nearby. So this very patient life strategy and the protective cyst coating around those eggs make it very difficult to kill. A lot of chemicals have difficulty penetrating that cyst shell. When a host crop is growing nearby, PCN will hatch. It'll attach to plant roots, and it'll feed, and eventually it will reproduce. Host plants exude a chemical signal that triggers PCN to hatch. And PCN doesn't move very much during its life cycle. It's primarily spread with the movement of soil on things like equipment or vehicles or seed potatoes. At high populations, PCN causes an 80% yield reduction, and to the plant causes wilting, stunting, and can even cause early plant death. PCN is considered a pest of major concern worldwide. It's a quarantine pest in over 80 countries, including the US. The initial detection of PCN in Idaho had a very negative effect on our trade of Idaho potatoes. Canada, Mexico, and Korea all shut off importation of Idaho potatoes upon hearing PCN was found here. And Japan even cut off importation of all US potatoes. Through the efforts of the PCN program to contain and delimit the infestation and also to work towards eradicating the pest, all of those markets have been reopened with the exception of potatoes from Idaho to Japan, and that's a work in progress. The PCN program regulates fields that have been, where PCN has been found, and also regulates fields that have been exposed to soil from infested fields. We also regulate the movement of certain articles off of infested and associated fields. Basically anything that has the capacity to carry soil away from a field is regulated. That's namely farming equipment, vehicles, you name it. We've dealt with just about anything coming out of a field that needs to be regulated. Any article that can be washed at the field to remove soil, that is how we deal with that issue. So there's a great effort to sanitize farm equipment at the margins of field just to return that soil back to the field of origin. Articles moving out of infested fields have to go through an additional sanitation treatment, which involves being steamed under a tarp for at least an hour. For articles that cannot be sanitized at the field, they're allowed to move under a permit with very special handling and containment and disposal requirements during transport and at the receiving end. Potatoes can be grown on associated fields, but unfortunately potatoes are prohibited on PCN-infested fields. And the reason for that is because potatoes are the thing that multiplies the pest. So we don't want to make the problem any worse. An infested field, once it reaches a point that it's gone through a series of very rigorous tests to demonstrate that there are no living PCN left in the field, then it is eligible to return to potato production. And at this point, after 11 years, over 70% of the infested acres that have been found are at the point where we can no longer find viable PCN, and those fields are eligible to return to potato production. So at current, we have 27 known infested fields in Bingham and Bonneville County. That's just over 3,000 acres worth. That's less than 1% of the entire potato production area in Idaho. So the program has been very successful with containment. We have sampled soil from across Idaho, all but one of potato-producing counties in the state, as well as survey efforts in other potato-producing states. And so far, this pest is found no place else in the country except for this small area in southeast Idaho. Program regulates another 6,200 acres of associated fields. Those fields are going through the soil testing procedures that are necessary to hopefully completely deregulate those fields within the next year or two. The PCN program has historically sponsored eradication treatments on infested fields. Those treatments started in 2007. The primary treatment that was used was soil fumigant methyl bromide, not your average soil fumigant in southeast Idaho. It was very specialized. It was something that required bringing a contractor, a chemical applicator over from Washington to do. It was extremely effective. One treatment with methyl bromide would reduce egg viability by up to 95%, a second treatment up to 99%. So things were going well with that eradication effort until in 2015, some concerns were raised about bromide residues showing up in certain crops grown on treated fields after treatment. So APHIS made the decision to discontinue use of methyl bromide for eradication of the pale cyst nematode. Since then, we've had some good options come down the pike. We have been treating fields with telone 2 or 1,3-dichloropropene. We have a special local needs label, or a 24C, that allows us to apply at a higher rate than what is commonly applied in this area. And we find it has a 50 to 60% kill rate with one treatment. Not quite as great as methyl bromide, but it doesn't seem to have any of the residue issues that we had with methyl bromide. Other pros for the use of telone are that it's a fraction of the cost of methyl bromide, and we're able to use local companies and local applicators to come and do those treatments on infested fields. So as I mentioned, at this point, 20 of the 27 infested fields are at a point where we can either detect no PCN or we only detect dead PCN. Over the last several years, the University of Idaho and the USDA Agricultural Research Service have done some really terrific research looking into a trap crop as another treatment for pale cyst nematode. They've been looking at Lychee Tomato, or Latin name Solanum cisimbrifolium. What is a trap crop, you ask? Trap crop is a plant that can mimic that chemical signal that a host crop emits that tells PCN it's time to hatch and feed and reproduce, except Lychee Tomato has a natural immunity to the pest, so the pest hatches, it's unable to establish a feeding site, and it dies before it can complete its life cycle. We've used Lychee Tomato on a production scale in infested fields since 2015, 130 acres in 2015, 50 last year, and we've got another 36 that are planted this year. And of the 3 fields planted in the last 2 years, we've seen viability, our detection of viable nematodes go to 0. And on one field, not only can we not find any dead PCN, we just simply can't find any PCN at all. So there's a tremendous amount of promise with this trap crop Lychee Tomato. Not my research, but I'll just touch on it briefly. Lychee Tomato seems to have an efficacy rate of up to 98% kill of PCN. It's not a crop that is harvested. It is simply grown for the purpose of treating for PCN, and then it's killed at the end of the growing season. And Pam will talk a little bit about that. U of I has had a number of research trials in our infested fields, and they've just been terrific to work with. This is a very exciting tool. We hope that the PCN program can scale up and to use this across more acres every year. What it will take to do that is to partner with a commercial seed producer. That's something that we're working on. One more thing I'd like to add is that through the last few years of using Lychee Tomato, we discovered there was a lot of misinformation out there about the plant. Folks saying it's a noxious weed, it can't be killed, you can't control it, and that's where Pam's research comes in. She'll tell you about that. None of those things are true. And the U of I has been really great with their public outreach campaign, especially this year, just to dispel those myths about the plant and to share the good news about it and how effective it is. And through those efforts, we've seen even our toughest critics of Lychee Tomato come around and support this for future use in Southeast Idaho.
To complement what Tina just said, it originated from South America, and it was introduced into Europe in the 1850s. And as you just heard about, in 2006, it was detected in Idaho. PCN dwells into the soil as a cyst, and as the potato root diffuse, which is the exudation from the potato roots, make contact to the cyst, that provoke egg hatchings. And once the eggs hatch, the J2, which is the second stage, the juvenile, migrate to the roots. And the J2s use its stylet in order to get inside the roots. Once inside the root, it establishes a feeding site where it continues with its life cycle from J2 to J3 and into J4. The fourth stage, juvenile, that's where the sex differentiation occurs between male and female. Eventually, the male will exit the roots and will fertilize the female. And that's the female that will continue molting into a cyst. And inside the cyst, you can find around 300 to 600 eggs. So that's the main component of the life cycle. So the cyst, as you heard about, can survive into the soil up to 30 years. At the University of Idaho PCN lab, we've been conducting research, for example, using trap crop, which is the Lychee Tomato. We found that the Lychee Tomato provokes egg hatching, but unlike potato, Lychee Tomato is not a host. The Lychee Tomato, what it does, it just disrupts the life cycle of the nematodes. And some result, we found that we have a huge PCN reduction up to 95% when we planted those, when we put those cysts on the Lychee Tomato influence, and then we plant it with potato crop. And also we're doing a research on using biological control agents to control these important pests. For example, we've been using Trichoderma asianum, and in our result, we showed that up to 98% reduction in PCN in greenhouse experiments. My colleague Cole Arder will explain you about biofumigant applications.
The mustard meal that we are working with specifically, or the plant, is Brassica gensea, otherwise known as Pacific Gold, and it contains a glucosinolate called sinigrin, and that sinigrin, when it hydrolyzes, it produces the chemical 2-propenyl isothiocyanate, and that is responsible for the kick that you taste in mustard, horseradish, and wasabi. And so the mustard meal has been formulated by one of our coworkers in agricultural science, and they have concentrated it down to a wettable powder, and when that is applied in the soil and wetted and covered with a vapor layer, it can fumigate the soil and eradicate the pale cyst nematode in the soil over the course of 2 to 4 weeks. We have conducted greenhouse and field experiments with great success. When we conduct those experiments, we treat them for an allotted amount of time and then perform hatching viability and then bioassay viability, where we plant them under susceptible potato plants. So after conducting those assays, we found that the pale cyst nematode, after being treated with Brassica gensea meal, showed no hatch compared to our control groups and no reproduction under potato. So it was largely successful in the greenhouse. We are conducting field experiments and have been for the past several years down in Shelley, Idaho at one of our field plots. And we are testing Lychee Tomato, we have tested biocontrol agents, and we are also testing Brassica gensea meal. So in the past we have done Lychee Tomato time trial tests to see at which point of growth the Lychee Tomato becomes effective at eradicating or attracting the pale cyst nematode to its roots. We've done that in the past 2 years. And then this year as well, we are conducting a time trial as well as trials with sterile lines of Lychee Tomato. So we have diploid, triploid, and regular lines that we're testing to see if they are still effective at eradicating pale cyst nematode while still being sterile. And the benefit of those sterile lines is that we don't have to worry about them reproducing in the field. So they're kind of a no-risk plant, and they can be planted and killed after the growing season without risk of spreading to other fields. Those are the 2 trials we have for Lychee Tomato, and then the final trial we have is testing the effective rate of the mustard meal biofumigant in the soil. So we plant a microplot field trial and apply the biofumigant, and after doing so, we take cysts from the soil and test them to see if the rates that we have applied are effective or not. And the purpose of these field trials, as opposed to our lab and greenhouse trials, is to see if everything that we're doing works with more variables, variables that we cannot control, weather, soil type, and a host of other things. So that is currently what we're working on, and yeah, that should be the bulk of our work this summer. And we are actually taking up our final experiment today, so for the biofumigant, and we'll see what the results are.
All right, as Tina Gresham from APHIS has mentioned, and our 2 graduate students from University of Idaho, we are working with a trap crop, Lychee Tomato, scientific name Solanum cicimbriifolium. Also, as Tina mentioned, there's been some misconceptions about this plant. Some people thought it was a noxious weed, invasive, and that it would take over our crop systems here in Idaho if we brought it in and used it. We do need to be careful with anything we bring into the state that is not native to the state, however. And so we go through a very stringent process to get a permit from the Idaho State Department of Agriculture in order to plant this as a trap crop, not only for research, but out in the grower fields, as we've been able to do the last couple of years. We do have, again, 1 grower field this year near Shelley, Idaho. It's in a quarantine field where the growers planted Lychee Tomato, and we have our test plots out there for herbicide management. If you turn on your tour book, there's a white page that's entitled PCN Trap Crop Lychee Tomato Fact Sheet. And we developed this so people would have a little bit more information about the trap crop. It's an annual, of course it's related to potato. It's native to South America, and it prefers warm soils and climate. And it is sold commercially as a trap crop in Europe. There are some seed companies, and one of them is in Germany, and they've used it in the Netherlands and in Scotland and some other places where they have pale cyst nematode. So Chuck Brown, our potato breeder with USDA up in Washington, thought, hey, let's bring it over here. Let's see if we can make it work for our pale cyst nematode here, because we just want to get rid of it. And if we can use a trap crop to do it, that would be a very good thing for us all. So Lychee tomato has been introduced into the United States. It's found in 17 states, southern part, eastern seaboard, and then up into Canada. And it's not listed in any state as a noxious weed, okay? So that's really important to know. It is grown, people do grow it in other places for the berries to make a jam and that kind of thing. So it's an interesting plant, but in Idaho, the only way you can plant it is if you get a permit from the Idaho State Department of Ag. And the one thing that's great about this project that we've been working with for PCN eradication is we have University of Idaho, Washington State University, Oregon State, people from New York, Cornell, International Scotland, working with growers here, the Idaho Potato Commission, USDA-APHIS, and many other entities, and it's really great to have these groups working together on this type of a project. It's a bad thing that happened in Idaho, but it's a good thing that we're brought together to work on it. So my work with Lychee tomato is to make sure that it does not become a weed, okay? And so I need to be able to tell growers how to kill Lychee tomato when they don't want it, and that way it won't become weedy. However, the other thing that's very, very important is to be able to control weeds in your Lychee tomato trap crop. And one of the ones that you really want to control is hairy nightshade or any of the other nightshades, which is a problem here in Idaho, because that weed can host the pale cyst nematode. So it wouldn't do any good to have a trap crop out there if you had hairy nightshade out there, because the nematodes will come over to the hairy nightshade and be able to survive into adulthood there. So my project since about 2012 has been to look at pre-emergence, post-emergence, and end of the season herbicides to control Lychee tomato and to also control weeds in Lychee tomato. And I have some information here on a few pages with some of the results that we've had since 2013 with pre-emergence herbicides. I also have some pictures for you. And we've had some really nice trials where we've shown a wide range of herbicides that will kill Lychee tomato, all right? We have only found a few herbicides that are safe to Lychee tomato. So that's been the real challenge, actually, is to be able to control weeds in Lychee tomato. One of those herbicides is Sonalan, and the other one is Matrix, and you can apply Matrix pre-emergence or post-emergence. So very interesting challenges to be able to control weeds in it, not to kill it. We're able to do that in all sorts of crops, non-crop situations with all the different herbicides that we've been able to find. Okay, the last thing I want to talk about today are these plants right here. Remember that people are concerned that it will become a weed. Well, our researchers up on campus, Joe Kuhl and Bob Terpepi, have been able to develop a sterile Lychee tomato, okay? So that means we can plant it. We don't have to worry about berries and seeds becoming mature in the field, falling to the ground and germinating at some point when we don't want them to. So there's another page here. It's called Sterile Lychee Tomato. Gives you some information about how, just some real simple background information about how Joe and Bob were able to develop this, along with Chuck Brown and also Louise Marie Danduran. The wild types that we've gotten from Europe and that we've grown in the field, they're diploid, okay? We can generate tetraploid plants. Those have 48 chromosomes. And when we cross the tetraploid with the diploid, we get triploid, which have 36 chromosomes, and those plants are sterile. So this has been a great project. The growers are really excited about this. Joe was able to get a couple thousands of plants that he's generated in the greenhouse and sent it down to us here in Idaho, and we've been able to transplant these the field that we have where we've planted, drilled in our lychee tomato, we've been able to transplant some of these as well. And what we'll be doing is looking at berry and seed production, but we know those seeds and berries, they are sterile. And so I was able to bring back about 100 plants here to the research station so that I could show it to you on this tour. So again, we're pretty excited about this. These plants can get up to 4 feet tall. They are not winter hardy. Frost will kill them, and they just do not overwinter in our region of Idaho. But having a sterile lychee tomato would be a great thing. So just to end then, we had Tina Gresham talk to us from APHIS about pale cyst nematode when it came to Idaho in 2006 and how they've been working hard to help eradicate it and the reduction in acres that we've seen in grower fields. We had our graduate students talk a little bit about their projects from University of Idaho. And then I've talked to you a little bit about the lychee tomato. We've gotten some really nice funding to be able to do this work from the Idaho Potato Commission. We also have some USDA grants. One of them is a 5-year grant. We're in the 3rd year right now. And through that grant, we've been able to put up a website called Global. And I have some handouts back at the research station where we have a newsletter generated and we posted on that website. And we have it not only in English, but we also have it in Spanish language as well, because a lot of people in Idaho speak Spanish and they're out in those fields, and we want to be able to relay to them the information that they need to know about pale cyst nematode.